Well, good evening, everybody. Welcome to Fox Chase. It's great to see so many of you here. It's a great turnout. My name is Frank Hoke. I'm Vice President for Communications here. We're delighted this evening to be able to host Jessica Wapner, author of the Philadelphia Chromosome. Today is the book's official publication date, so tonight is special. It's something of a book party to celebrate the book and Jessica. From here, I believe she heads up into New England and on to stops across the country on her book tour. So congratulations and good luck. Should be a lot of fun. Jessica's book tells the story of the discovery of the Philadelphia Chromosome, of course, more than 50 years ago by two men, David Hungerford here at Fox Chase and Peter Knoll at the University of Pennsylvania. The two men co-discovered the chromosome. And the book tells of that discovery and also the subsequent many years of scientific and clinical research that followed that discovery. We are particularly honored tonight to have members of both men's families in the audience, and I won't ask you to, raise your hands or stand or anything, but it's really gratifying to have members of the Knoll family here and members of the Hungerford family. We're very happy to have you. As you'll hear, it took years of hard work, but the discovery of the chromosome led directly to the development of one of the first targeted therapies for cancer, a drug called Gleevec. The generic name is imatinib. Gleevec changed a deadly blood cancer, chronic myeloid leukemia, into a disease that can now be clinically managed, with most patients living many years with their cancer controlled by the drug. So it was a real watershed discovery and process, and the drug at the end of that process. The chromosome, the Philadelphia chromosome, was the first genetic anomaly found to cause a form of cancer, but we now know that cancer is always a disease of broken genes. And so targeted therapies like Gleevec really represent the future of cancer medicine. And that's the understanding that guided the founding of Fox Chase's Cancer Genome Institute just a few months ago. With a unique 45-gene panel, we can now test a patient's cancer for the genetic signature of that tumor and increasingly offer targeted therapies to treat the disease. These are early days in this new approach to treating cancer, and we still rely on many of the other very excellent treatments that have been developed over the many decades and are available to us now. We do a pretty good job these days, I think, of helping people with cancer. But the story you will hear tonight does indeed point to the future of cancer medicine in a new way. For some of you, this may be your first visit to Fox Chase, and I thought I'd give you a brief overview of who we are before we introduce Jessica. First, I think our mission tells you most of what you need to know about us. The mission of Fox Chase Cancer Center is to prevail over cancer, marshaling heart and mind in bold science, breakthrough medicine, and personal touch. You'll also want to know that we're a federally designated comprehensive cancer center. We're one of only 41 in the country with that designation. Comprehensive in this case means that we consistently integrate research into the care delivered here at every level of what we do. It also means that the comprehensive tag also means that we address the challenge of cancer at every stage, from risk assessment and prevention through survivorship, and support patients in every dimension of the cancer experience, body, heart, and mind. A few other just quick facts about Fox Chase. Our scientists have won 2 Nobel Prizes, among many other top scientific honors. We're very proud of that. 1 in 4 of our physicians are listed in the top docs issue of Philadelphia Magazine every year, which is a remarkable percentage in terms of just the quality of our clinical faculty. Our nurses have earned the highest honor in their field. It's called magnet status, 3 times in a row, and only 1 in 50 hospitals in the country can make that claim. And we're also routinely ranked as one of the top cancer care facilities in the nation by U.S. News and World Report. Lastly, this past July, Fox Chase Cancer Center became a member of Temple University Health System. And this affiliation with a major academic health system is already expanding our caregiving capacity and capabilities and our research horizons in many important ways. So we're really looking forward to seeing how that develops and unfolds for us over the coming years. About 2,400 staff and faculty at Fox Chase serve more than 30,000 patients and their families every year. We're very proud of what we do. I'd like now to introduce you to Dr. Joe Testa, who will introduce our guest of honor tonight. Dr. Testa holds the Carol and Kenneth Wegg Chair in Human Genetics and has made a number of significant discoveries in cancer biology and genetics. In 2011, for example, he co-led a major study that identified a gene linked to mesothelioma, which is a dangerous cancer of the chest wall and cavity, often caused by asbestos exposure. Dr. Testa was also a postdoctoral fellow in the laboratory of Dr. Janet Rowley, who played a pivotal role in the scientific story of the Philadelphia chromosome, and I suspect we'll be hearing a bit more about her later, and the development of Gleevec. So he knows tonight's story very well. Joe.
Thank you very much, Frank. So Jessica Wapner is a freelance science writer. I see we have Gwynedd Mercy Academy students here, and that's exciting for me. My daughter is in 11th grade and starting to think about colleges and what field of career she'd like to enjoy, and I think it's going to be medicine, and I'll bet there's some in this audience that feel the same way. So I think if you want to consider Fox Chase for its summer program, you might want to look into that. It's a little plug for them. They have a wonderful setup this year where about 150 students from the area are coming to Fox Chase to learn about all the different disciplines of medicine, nursing, every field that could help in a cancer center, like librarians and so forth. So you may want to look into that. So as I said, Jessica focuses on biomedicine and cancer, but she's also written about things like viruses and so forth. Her works have appeared in many prestigious venues, such as Scientific American. I'm still waiting for my first publication in that journal. She's got about 9 or 10. She's also published articles in the New York Times, Science Magazine, The Scientist, and Nature Medicine. She also writes for PLOS, Public Library of Sciences blog on the ethics and economics of drug discovery and drug development. She belongs to a rare breed of talented writers who's able to succeed in making medicine understandable to the public. There are not many of them, believe me. I have been misquoted more than once by those who are unable to convey the message you're trying to present. Jessica is a very passionate spokesperson for biomedical science. For example, she recently wrote a thoughtful opinion editorial for the Los Angeles Times entitled, "Don't Cut Life-Saving Dollars. Health research and medical progress are in peril amid NIH budget cuts." So obviously she immediately, in writing this timely piece, earns instant fans among cancer researchers who are all facing daunting federal grant pay lines and budget cuts. I'm not sure many of you are aware of it, but right now, and the students shouldn't listen to this part, only 1 in about 12 to 15 grant applications will be funded. By the time you're in, you have your PhD or MD, it's going to go way back up again. Thanks to people like Jessica. So this evening we're delighted to have Jessica speak to us about her new book, The Philadelphia Chromosome. Interestingly, the subtitle is A Mutant Gene and the Quest to Cure Cancer at the Genetic Level. That's actually very, very meaningful because I want to point out to you, cancer by its very nature is a genetic disease. There are 2 kinds of genetic alterations that occur. Some of them you can be born with. Some people have heritable mutations in genes such as BRCA1 or BRCA2. For example, today I heard Angelina Jolie is going to have prophylactic removal of her breasts and reconstruction because her mother died of breast cancer and she inherited the BRCA1 gene, I believe. And another Miss America, I think it was, came out today and said that she's inherited the BRCA2 mutation in it to decrease her chances of getting cancer, which are 87% for women who have this mutation. She's also going to elect to have breast removal and reconstruction. Another kind of genetic alteration occurs during a person's lifetime. These are called acquired genetic changes, such as the Philadelphia chromosome. So that chromosome way up in the top there is a normal chromosome 9. The one right below it is the abnormal 9 that has a piece dangling out to the left. And the chromosomes at the bottom, the second one up is the normal chromosome 22, and the one at the very bottom is the Philadelphia chromosome, which is smaller than its partner, and a piece of that chromosome has gone translocated up to chromosome 9. So this is from a person who inherited normal chromosomes, but sometime during the person's life, got this rearrangement to occur. It might have been because of an environmental exposure. I study mesothelioma, where these mutations are thought to arise because of exposure to asbestos. Some skin cancers, you can have exposure to sunlight that can lead to cancers and so forth. In preparing her book, Jessica interviewed more than 35 people who played a direct role in this epic journey that unfolded over nearly 5 decades, starting in 1959 here at Fox Chase and at the University of Pennsylvania, and it was called the Philadelphia Chromosome because it was found here. In addition to Jessica, we are honored to have the justifiably proud families of David Hungerford and Peter Knoll, who were able to join us for this evening's special event. Their relatives are superstars in the cancer research field and people who we are justifiably proud of as well here at Fox Chase and at University of Pennsylvania. I like this story because together, a basic scientist, David Hungerford, and a physician, Peter Knoll, came together to work on a project that neither one of them would have been able to do alone. It was this partnership, this collaboration that allowed them to do great things. What they did was they discovered the first consistent clonal chromosome abnormality ever associated with any kind of human cancer. The discovery of the Philadelphia chromosome more than 50 years ago was accomplished with rudimentary tools during a much gentler era. If you walk out of this room, you'll see the original Philadelphia chromosome microscope that David Hungerford had used, which was from the 1940s. And it's pretty archaic by today's standards, but we were able to find the Philadelphia chromosome in a metaphase, if you want to take a look at it on your way out from this room when we're finished here. It's a miracle. You think they started, and it's very hard for me to even see the chromosome that David and Peter observed. So how they did this was just keen observation. Nowadays, things have changed so much, and a lot of these tools are allowing us to look at every gene in the genome, 22,000 genes at one time, to find the genetic abnormalities that are present in the cancer. Anyway, this genetic discovery here in Philadelphia would lead to a frenetic race to find the critical gene that was mutated as a result of this Philadelphia chromosome and to design a potential cure. So Jessica's book helps us to fully appreciate just how hard-won and consequential were the achievements that she chronicles. Her book gives readers an appreciation of the principles behind some of today's most important cancer research as doctors and scientists work feverishly to design novel drug therapies that can attack the genetic roots of each tumor individually. That is, the notion of personalizing cancer care based on targeting a specific genetic alteration present in a given tumor or a blood disorder such as leukemia. We know what the genetic abnormality is, what drug works for that genetic abnormality. That's the goal. Jessica's book has already received outstanding reviews. Publishers Weekly called the book a gracefully written history that skillfully combines both the science and humanity of a fascinating search for a cure for CML, or chronic myeloid leukemia. A book review in The Scientist stated that the way Wapner repeatedly adds up preceding steps to build to the scientific breakthrough is masterful, making for compulsive, surprisingly emotional reading. Sid Mukherjee, the author of the Pulitzer Prize-winning book, The Emperor of All Maladies, said the following about Jessica's book: "It is truly the story of modern cancer biology. Jessica stitches the whole story together with tenacity, diligence, and humor. This is a wonderful, readable, and highly informative book." It is my pleasure to introduce this evening's special guest, Jessica Wapner.
Hello. Hi, thank you so much. Thank you for the lovely introductions, and great to see everybody. It's so wonderful to be in Philadelphia. This is the city where my research started, and now it's wonderful to be back here on the day when the book is published. I am going to ask the relatives of David Hungerford and Peter Knoll to please raise their hands, because I think we should know the fame we're sitting next to. So I will gradually migrate away from the podium, but I'm going to begin the talk with an excerpt I want to read to you from the book. What I'm going to do in this talk is I'm going to read you an excerpt from the foreword of the book. So I'm not giving anything away. I'm just reading you from the prologue. And then speak about my journey, my experience with researching this story, and how I came to feel that this needed to be told in a book, what is significant about this story, why it matters to me, which might be also why it matters to you. And then we can have a Q&A for a little while, right? There's a little time for that. So I'll speak for about 20 minutes. We'll have the Q&A, and then there's book signings. So, all right? Great. Okay, so I'm going to begin with an excerpt. Okay. So this is from the prelude, which is entitled Down to the Bone, February 2012. Gary Eichner sat in a chair backed up against a wall. Across the room, his nurse was half hidden by a computer. She scrolled through his medical chart on a monitor he couldn't see, typing his responses to her questions. He answered and smiled, as if he were just fine, as though that would somehow make it so. He kept his worry concealed, silently wondering what the next hour would reveal about the disease that had so suddenly overtaken his life. Fear permeated his every thought. The nurse ran through the litany of side effects that he might be experiencing from the leukemia medication he'd recently begun to take. Any chest pain, she asked? Heart problems? Any swelling in your ankles? Any problems with nausea? Eichner's blanket no was tempered only by his description of what happened when he took the drug on an empty stomach. It's massive cramping, massive pain in the stomach, he told the nurse. It's just like the worst thing you've ever had. The nurse knew that Eichner really had no idea of the worst thing someone with his type of leukemia could have. Few patients with Eichner's disease today will ever know that kind of pain. But if the nurse was thinking anything of the sort, she kept it to herself. After all, her patient was about to have a large hollow needle inserted into his bone so that his doctor could extract a sample of marrow. Having witnessed countless such procedures during the 12 years she'd been a nurse to patients taking the drug, she knew how much was riding on the biopsy, and she knew that Eichner, age 43, was well aware that his life was at stake. If anyone could set him at ease about that, it was his doctor, Brian Drucker. All I care about is that you're making some progress, Drucker told his patient, who had come to Drucker's clinic in Portland, Oregon, for a bone marrow biopsy. The procedure would reveal whether the medicine, a pill he'd been taking daily for 6 months, was tackling the leukemia that had invaded his body. Eichner had chronic myeloid leukemia, or CML, a cancer of the white blood cells that, though slow-growing, could be fatal. I will be happy if you're 18 out of 20. Eichner nodded his understanding, his jittery foot the only sign of his nervousness. For Eichner, those numbers were part of the new language he'd learned since his diagnosis in the summer of 2011. As with so many cancer diagnoses, the education began suddenly and unexpectedly. After a day or so of sharp, excruciating kidney pain, Eichner, a single parent of a teenage boy, drove himself to the emergency room at his local hospital in Olympia, Washington, where he'd been living at the time. His sister-in-law, a trauma flight nurse, had told him she thought he had kidney stones, so Eichner was expecting to be admitted for a standard, albeit painful, procedure. But when several doctors walked into his hospital room together, he knew something wasn't right. "You don't have kidney stones," they told him. "You've got what we believe is leukemia." A blood test soon revealed that Eichner, in otherwise perfect health, had CML. The excess number of white blood cells contained in the sample confirmed that much. The doctors explained that the stabbing pain he thought came from his kidneys probably was from his spleen, enlarged by the high concentration of leukemia cells within. Although the slow-moving nature of the disease meant he wasn't in immediate danger, there wasn't any time to lose. For the best chance at long-term survival, he needed to start treatment right away. As Eichner was rapidly informed, danger could eventually come. If the treatments didn't work, within 5 years his bone marrow would fill with blast cells, white blood cells that fail to mature, and thus are both overly abundant and thoroughly useless. His blood, once free-flowing, would turn into viscous sludge. The supply of iron-rich red blood cells that carry oxygen around the body would steadily plummet, leaving him fatigued and anemic, while a decrease in the number of platelets would render his blood unable to clot. As the disease progressed, the minuscule capillaries leading to his eyes and brain would clog. His spleen would likely become profoundly enlarged. As his body began shutting down, he would bleed in his brain, in his intestines, and out of every orifice. 2 days later, Eichner was having his first bone marrow biopsy, performed by a nurse who spoke broken English and had to climb on top of Eichner to hammer in the 4-inch long needle to break through his bones, which had become hardened and inflamed from the profusion of white blood cells inside. Finally, she managed to extract an ounce of marrow, the spongy matter in the center of our bones where new blood cells are made. In the genetics laboratory, a fluorescent dye applied to the coiled strands of DNA inside Eichner's blood cells revealed the telltale sign of CML, a genetic mutation known as the Philadelphia Chromosome. An abnormal chromosome regarded as the defining feature of this fatal, spontaneously arising cancer, and discovered more than 50 years ago. 20 out of 20 cells in a sample of his marrow contained this genetic error. A family friend with the same type of cancer told Eichner to call a doctor named Brian Drucker right away. Don't do anything before you see him. The friend told Eichner a couple of days after his diagnosis. 3 days later, Eichner got a call from Drucker, who'd mysteriously, Eichner didn't know exactly how, gotten his phone number. During the next 20 minutes, Drucker talked Eichner down from his panic, assuring him that, being in the early stages of the disease, Eichner could wait a week or two before making the trip south to Oregon Health and Science University, where Drucker had been treating and researching leukemia since 1993. He gave Eichner his professional blessing to go drink beer. It was August, and Eichner had been getting ready to leave for a day at Olympia's annual Summer Brew Fest, his brother's idea for getting his mind off leukemia for a while, when the phone rang. 10 days later, Eichner was on his way to Portland. Within 3 weeks of his diagnosis, he was swallowing his first tablet of medication, which Drucker had been instrumental in developing, a drug aimed at tackling the cancer at its root. So let me turn on this mic so I can move around. Okay, is this, yep? Hi again. So that was a lot about Brian Drucker, who's in Portland, and I'm going to talk a little bit more about the Philadelphia connection, of course. Is this okay? Am I too echoey? I'm too near the microphone. I can't. Too near this microphone. Okay. Is that better? Is this one still on? Okay. We're in business. So I won't keep you too on the edge of your seats, Gary Eichner is still alive. And his test results, I'll tell him you applauded. This is in the foreword, this is in the prologue, so I'm not giving anything away. His test results come back as having 0 out of 20 cells positive for the Philadelphia chromosome. So he started out with 20 out of 20, and 6 months later, he's 0 out of 20. Yeah, he's the dream patient for this drug. But there are many, many dream patients. Almost all patients are dream patients for this drug. So why I wanted to start off with this excerpt is, to me, it encapsulates so much of what is just incredible about this story. I mean, first of all, it's very clear we see the impact that scientific progress has on our lives and the length that we go to ensure that progress happens. One of the most stunning things to me about this story is just how much it exemplifies this whole story, not just Gary Eichner's story, this unstoppable drive that we have to make tomorrow better than yesterday, and that people are willing to fight tooth and nail to make that happen. Of course, I didn't know that when I started out researching this story. I first learned about the Philadelphia chromosome when I was working at a medical journal around the year 2000. And those words were enough to make me wonder, what's the Philadelphia chromosome? I mean, what a great name, first of all, for a genetic mutation. I mean, BRCA, you know, it's so much harder to remember. They should all be named something captivating like that. I didn't know too much about it. I knew that it was connected with CML, which is a rare disease. It's diagnosed in about 5,000 people in the US each year, or 1 or 2 per 100,000 globally. So as many as 75,000 people worldwide can be diagnosed with this disease. Left untreated, a person would die within 4 to 6 years. And in fact, that was often the prognosis before the advent of Gleevec, which is the drug that is discussed in this book and which I'll get to in a little bit. When I moved from working at a journal to writing for a mainstream audience about science, one of my favorite things about science writing is just keeping an ear to the ground for story ideas. And my mind turned back to the Philadelphia chromosome. What was this about? And truth be told, this book started with asking about the name. I called up some people that I knew, Brian Drucker, some other scientists, and asked them, well, where did this come from? And I found out that it had been discovered at between the University of Pennsylvania and Fox Chase Cancer Center by Peter Knoll and David Hungerford. And as Alice Hungerford explained to me over the phone back in 2007, that her late husband he was this unstoppable observer. He was this old school scientist who just loved to look at things. And teamed up with Peter Knoll, as Dr. Testa described, who, as he explained it to me, he was just going to lick this cancer problem. He was young and thought he could pretty much do it over a summer, as I talk about in a book. And that's what they were doing. They were looking through microscopes. And one day, David Hungerford is looking through a microscope using this technique that Peter Knoll had stumbled across that allowed them to look at chromosomes in a way that was just more expanded, a more expanded view than anyone had gotten to at that point. And one of the chromosomes was too small. That was in 1959. He snapped his shutter on the camera, took the picture, and that picture was the start of what would become decades of research, unraveling the connection between this genetic mutation and CML, and that would eventually lead to the first drug to target cancer at its root cause. There are so many twists and turns in this story, and as I started talking with people about it, so I came to Philadelphia in 2007 to meet Peter Knoll and heard his amazing story. he had gone, right when he was getting interested in researching cancer, he was drafted. So he had to wait and come back, and then he really got into it and met up with David Hungerford. It's such a serendipitous pairing of those two, that they happened to be in the same city, these two people who were perfectly equipped to find this discovery. But then the more I asked, the more it was, well, wait a minute. I thought maybe it's going to be a magazine article. Maybe nobody has told this story yet. But then every time I asked another question, there was this whole flood of research that would come. So to give you an example of some of the science that you'll read about in the book, the Philadelphia Chromosome encodes a protein that is called a kinase. We have many kinases in our cells. The kinase encoded by this genetic mutation is one that triggers the production of white blood cells. Normally, white blood cells are sometimes being produced and sometimes not. But in its mutated state, the body, inside the bone marrow, there always is the signal to produce white blood cells. So I asked the question to somebody, what's a kinase? Where did that come from? That discovery went back to the 1970s to a protein and a gene called SARC, which was short for sarcoma. So there was a man named Ray Erickson who was studying at the University of Colorado, trying to understand what SARC was about. And so I asked Ray Erickson, well, what was SARC? Where did that come from? And he said to me, well, that came from a virus that had been extracted from a chicken, from the Rouse sarcoma virus. Well, where did the Rouse sarcoma virus come from? This came from a man named Peyton Rouse, who was studying cancer back in 1910. A woman had come to him, it's such a great story, a farmer, carrying her hen into his laboratory. The hen has a tumor sticking out of its belly. And she says to him, "Can you operate?" He said yes. This was at Rockefeller University, which is on the Upper East Side of Manhattan. He said yes, and we should all thank this farmer, okay, because her hen was infected with a virus that caused cancer. And what was important about that virus was not that it caused cancer in chickens, which is very interesting. It's not to knock chickens. It's very interesting, but that research over decades eventually led to the understanding that our healthy genes can mutate, and in that mutated state trigger cancer. And that was because the virus contains a gene that it had picked up from the chicken genome. Way back in history, nobody could say when. And when that gene got picked up by the virus, it mutated, and it was that mutated gene that was causing the cancer. The virus could infect chickens without causing cancer, but once it got this gene scooped up into its genome, then it started causing the cancer. So I'm talking with Ray Erickson and all of these people thinking, like, what is this story? How is this possible that there's the kinases there? A man named Philip Cohen in Scotland is pioneering the research of kinases and what they do in our body. Ray Erickson is uncovering what this one kinase is connected to cancer. He finds out that this gene, SARC, that Peyton Rouse had identified in the virus in 1910, that was a kinase. And then that leading eventually to Bishop and Varmus, through roundabout research that you'll read about in the book, uncovering the genetic underpinnings of cancer. When Peter Knoll and David Hungerford spotted that mutant chromosome, the connection between cancer and genetics was not like it is today. It was, I don't know, maybe. Nobody knew where cancer was coming from. It was just as devastating a disease then as it is now, but nobody knew anything about it. And it took decades of research that could so easily not have happened. Would any farmer walk into a university setting today and have her chicken operate on? Can you imagine the paperwork? Just the paperwork alone, right? okay, would be enough. I mean, she'd be like, forget it, I don't need the chicken, I'll live without it. So the science and the medicine was enough to make me think, you know, there's just too much for one article. This has to be a book. But then the story takes a whole other turn, because once the picture became complete, that the Philadelphia chromosome encodes a mutant kinase, and that mutant kinase triggers the production of white blood cells, the unchecked production of abnormal white blood cells that fail to mature and fill the body up, as I described in the prologue, and leave no room for the healthy blood cells. As that picture became complete, that was in the 1980s, 84, 85, the very final proof came in 1990, that this gene and this gene alone was enough to cause cancer. But as it was becoming complete in 1984, 85, Brian Drucker and some other people felt they had enough to say, let's target this. Could we make a drug to block this protein, to throw a wrench into the clockwork and just stop it in its tracks? That's where the story just becomes this epic saga to make this drug. When I spoke with Brian Drucker in 2007, I mean, so many times, he was so patient with my emails and phone calls and meetings. I spent 3 days interviewing him in Portland last February, asking him, well, what happened? Because in 1984 was when Ciba-Geigy, a drug company in Switzerland, first started their program, their kinase inhibitor program, right? So these drugs, Gleevec and others that have come since then that target kinases are what are known as kinase inhibitors. It was 18 years between the time that they started the research program and the FDA approved the drug in 2001, which is a long time. I mean, granted, considering this research goes all the way back to 1910, 1959, maybe it doesn't seem so long, but in the world of drug development, it's a long time. And Brian Drucker, you know, he was instrumental in getting this drug made, and CML patients around the world credit him with making sure that this drug Gleevec got into clinical trials and got to them. And they credit him with saving their lives. And I wanted to know more about that. I wanted to know what took so long. What was the problem? Was there a problem? Was this just how drug development goes on and it was the process? What were the hurdles that they met? And you know how it is. You want to know, you get interested in something, you just want to know every corner, every little aspect of it. And so there were early lab tests, and what I began to learn was that there was the science and the medicine of the story, and then there were the people of this story. That not only were there the stories of Peter Knoll and David Hungerford and everybody who came before and after them that had a role in this, there were the people who were dedicated their lives to making sure this drug was going to get to patients. So as I began speaking with people at drug companies, I just spoke with as many people as I could to understand what had happened. People in the drug companies, people in academic settings who were researching the compound early on, patients, some of the first patients to enroll in the clinical trials. And what I found out was that there were two holdups with this drug. And this was part of another reason why I wanted to tell this story, because it pulls the curtain back on how drugs get made, on how new medications reach us. One of the reasons why it took so long for this drug to get made was because it was entirely new. I told you in 84, 85, people are saying, hey, can we make a drug to target this kinase, to target this haywire kinase? That was a completely new way of treating cancer. We all know about chemotherapy. I bet most of us in this room know somebody who's been through chemotherapy. It's a harsh treatment, and because it carpet bombs the body. It's general. It's not targeted. It just chemo means poison, so it's poison therapy. So essentially, you are trying to kill the cancer cells before you harm the person too much for them to tolerate the drug. What people were starting to think about as the picture between the Philadelphia Chromosome and CML became clear was called rational drug design. It was, well, we know the cause of cancer. We know why this is happening. So let's make a drug to stop it. Let's make a chemical that can get inside the cell and block this mechanism from happening. So Ciba-Geigy in Switzerland starts their kinase program. And they get about 5 compounds. It takes a couple of years, and eventually they have about 5 compounds that Brian Drucker in Portland is testing out. And he sees right away that one of the compounds is killing cancer cells in his laboratory, in a petri dish. What the company did was they sent him the 5 compounds. They knew which one was their most likely candidate. They had their lead candidate that they thought was the most likely candidate to be a drug. But he didn't know. They didn't tell him. They labeled them A, B, C, D, E. So he's testing them out. One of them wasn't even a drug, a compound. It was just a placebo, whatever it was made of. So one of them is killing cancer cells. Yes, it was the one that they thought was their lead candidate. And so he's in Portland saying, okay, animal studies, let's go. That's the next step. Drug company, right? The drug company is a business. Any company that sees that this compound is killing cells in a laboratory is going to be very interested. But they weren't going into animals right then. It was taking so long. Poor Brian Drucker was like, come on, guys, come on. What's the holdup? So as I said, it was risky, and it was considered a risk because it had never been attempted before. And as Jörg Zimmermann, who was the chemist that created the compound that we know today as Gleevec, as he explained it to me, he said, look, you have to understand, the shadow of thalidomide was hanging over the entire pharmaceutical industry, and still does. Right? I mean, this was a drug that was supposed to help women through their pregnancy and ended up deforming their babies. Nobody wanted another thalidomide on their hands. And so he says, yes, it took a long time to get through that, to get to the animal studies and to get through the animal studies. And so you can say on the one hand, why didn't you get it to patients faster? But on the other hand, well, if we had gotten it to patients and patients had started dying, well, you would be saying, well, how could you do that? How could you have taken the risk? So they were nervous, and they wanted to take their time. But that wasn't the only problem. The other problem was money. As I mentioned earlier, CML is a rare disease. It's diagnosed in about 5,000 people per year in the US. To give you a comparison, breast cancer is diagnosed in about 230,000 people in the US each year. To give you an even bigger number, about 1.6 million people in the US are diagnosed with cancer each year, any type of cancer. So the patient population of CML, or as it's talked about in drug company lingo, the market for the drug, was too small. How was this drug going to recoup on its investment, let alone make any money? So, which maybe shouldn't have been surprising, it was surprising to me. And because how did this drug languish in a pipeline while patients were dying of leukemia? It was in animal studies for several years. It was tested in mice and dogs and rabbits and monkeys, hamsters, and other kinds of cells. And it wasn't until 1998 that the first clinical trial started. it was not easy to get over the financial concerns, for sure. And it was only with, it's such a great example. It was people fighting for the drug inside the company, people fighting against that. Alex Matter, who was another chemist, cancer researcher inside Ciba-Geigy, told me that at one point during the animal studies, one of the people doing the animal studies said to him, "Not over my dead body will this compound go into man." So you can imagine that the fears, the concerns, and that was just from the biological side. They were worried that it was going to hurt people. There were liver problems in dogs, and the company said, "No, liver problems, people can die of liver problems." Brian Drucker said to them, "But we know the signs of liver problems. When you're in a clinical trial, you're under the most careful scrutiny imaginable. If we see liver problems coming up, we're going to stop the drug. We're not going to keep going, escalating the dose like you did in the dogs. We're going to stop the drug." So it was years of this going back and forth, Brian Drucker going to the FDA, all kinds of stories that finally got the clinical trial launched. That wasn't the end of the saga. There was more to it, because once they saw these stunning results, what happened was the drug was tried out in a phase 1 study, and patients were responding, in a way that was just unprecedented. Absolutely unprecedented in an early clinical trial, which is only meant to test out the safety of the drug. Patients are being kind of returned to normal. And so then the investigators are saying, well, we have to get this to everybody with CML. And the sickest patients, the healthiest patients, you have patients being brought in on wheelchairs with oxygen masks, being given this pill. It was a pill that you take once a day. And a couple of weeks later would be walking out of the hospital, dancing out of the hospital because they were so happy. And patients did play a huge role in this. I tell the story in the book of Susan McNamara, who in the months before she started being treated with Gleevec, lived between her bed and her computer. That was as far as she could get. She was so weak and so sick from the medicine that she was taking to treat the CML, which before Gleevec was a drug called interferon, which had incredibly devastating side effects. And she started a petition online to get this drug into expanded clinical trials. And to petition the company, which by then was Novartis, to make this drug, which she succeeded. So it was all of these things coming together that said, this is much more than an interesting story. This is a story that equips us with the knowledge to talk about healthcare, to talk about drug development, and to understand this current moment in cancer research, about why we're interested in personalized medicine in the first place, why we think about getting our genome sequenced, should we, shouldn't we, and where cancer research is at now. The FDA, when the FDA approved Gleevec, it was the fastest approval ever. This drug stands as one of the most successful drugs ever made for cancer, and has an incredible emotional story behind it about people, not just a miracle drug, but the miraculous lengths that people go to make sure that something that they believe in gets a shot in this world. So I wrote the book. And I will, thank you. Thank you. So do we have some time for questions? I hope a little bit, yeah? Okay. Are there any questions? There might not be any questions, yeah?
Is more money in the near future being put into targeted therapies?
Absolutely. So,
Relative to history,
yeah, definitely. So first of all, to address the Gleevec, the reason why the answer to your question is yes, is because Gleevec turned out to be a blockbuster, right? The money concerns stopped the drug in the pipeline for a very long time. But once the drug was approved, over the next 10 years, it was about $27 billion in sales. $27 billion. So they earned back their money in the first 2 years, pretty much. The kinase inhibitor market is about a $15 billion a year market right now. There's at least 15 of these drugs approved. There's about 100 more in development. And there's more to target than just a kinase, right? Because what this story showed was that if you find the underlying mechanism driving the cancer, then you can try to find a drug to block it. So whatever that underlying mechanism is. So yes, it is a huge industry, for sure. Yeah.
At the time, I guess it was in the 90s, Jesse Wapner died in a gene therapy situation. And how did that kind of correspond with the events that were happening here?
I don't know that story, actually. Do you want to, is it something?
I'm not the most informed in this area. I just know that it was
It's a gene therapy story.
Okay.
Not really a drug per se, but it was clinical trial information.
Okay. So, I mean, gene therapy, it's, in my mind anyway, it's different. I don't probably know enough to comment on it, but I think of it as a different, it's not drug development, it's altering the actual gene. So I would say look at them separately for now because it's not the same as blocking it, right? Yes?
Do we have any further understanding of possible causes of the mutation that leads to the Philadelphia chromosome, perhaps environmental causes, or is it a mystery?
As far as the current science knows, it's spontaneous. We get spontaneous mutations all the time, but we don't know about them because they don't make us ill. A mutation can arise and disappear. It can arise and not hurt us. But in about 5,000 people in the U.S. each year, a mutation arises that triggers CML. Whether there's any environmental trigger for it has not. If there is one, it hasn't been identified, and there have been efforts to link it. Yeah, I think, yes? What surprised me the most? So many things. So many things. I think one of the aspects that surprised me the most was how everything was so perfectly timed. I mean, even I talked about Susan McNamara. She only heard about Gleevec because of internet chat rooms. So this was so perfectly timed with, CML is a rare disease, so you're not going to find that many people in your hometown to commiserate with, right? So people were turning to the internet. This was the start of that, of people going online to find their community online. And so can you imagine, you're dying and you're reading online every day reports from patients on a clinical trial who are feeling great. They have the same disease as you, they've been through the same treatment as you, and they're feeling fantastic. And she says to herself, because she was told by Brian Drucker, she calls him up, she emails him, I think, to get on the trial, and he says, well, it's delayed. So I'll put you on a waiting list. But he had a waiting list 100 patients long, and he's basically having to decide who's sickest each time. And she says to me, she said, there was no way there was a drug out there that could help me that I was not going to get. Okay? But she only knew about it because of chat rooms. So, so that kind of thing is like amazing to me. Amazing. Exactly, and there's so much of that. Yeah. Yeah, it's a great name. Like I said, I'm from New York, but I love Philly. I love Philadelphia. So yeah, no, I was so pleased that it could have the name in the end. I'm going to go back here first and then to you. Behind you first and then to you. Yes.
What is the percentage rate of this, the success rate, percentage-wise?
So in the early phase 1 clinical trials, some of the response rate was 100%. 100% of patients were having their blood counts return to normal. What happens over time is that the disease can stop responding to the drug, and then people try. There's second and third generation Gleevec now. I think that the 10-year survival rate was, I think, more than 50%, which, it would have been 20% before Gleevec. And by more than 50%, I mean, I'm thinking well over. I have to get the most recent number. But it's, it's huge for a cancer drug, absolutely huge, yeah.
My question and statement is, I believe that every form of cancer is curable regardless. My sister had it. She was cradled like a baby. That's in the 1940s. And she was a miracle baby.
She had ovarian cancer.
She had ovarian cancer.
Wow.
And she came through it.
Right.
And she was a miracle baby.
Amazing.
And I say that anyone that has cancer, that knows someone that has cancer, it is curable by doctors like you.
Well, I'm not the doctor. I'm just the reporter. Thank you, though. but I agree with you. I believe that, too. I believe it is curable. I don't know that it will happen in our lifetime, that we will see a pill, a Gleevec, for every cancer. But I believe that we're at the start of that, and that we can say we were there then. Definitely.
Thank you.
Yeah, thank you. Thank you. Are we? Yes.
Other than leukemia, have the studies or trials on non-Hodgkin's lymphomas?
On non-Hodgkin's lymphomas?
Yeah,
I don't know if there's kind of, that disease is driven by kinase inhibitors, but there are a lot of drugs for NHL, so non-Hodgkin lymphoma, that are very close to curative for many patients. Yes.
What was the hardest thing to find out, either because people didn't want to answer questions, or people were hard to track down, or what was the kind of piece of information that you were very excited to get because it was hard to
That's a great question. I always wanted to know how much a monkey cost. And I'm never going to know. That I don't think is information that will ever be divulged. Well, I want to know how much does it cost to research a drug. So, how much do monkeys cost when you're studying a drug? The information I was most excited to get, really, I have to say all of it. It's hard to choose. I was excited to learn that Brian Drucker had a love story unfolding with all of this research at the same time. I had dinner with him and his wife because his first marriage had fallen apart, and you always want to weave in a good love story. So there was one. And the role that Susan McNamara played. I think every little piece to me was, one more reason to write this book. Was there someone else?
Thank you so much. I'll join me in thanking Vanessa for